So it has been really great the last couple of days. And now she's here for 2 weeks. Oh no. I'm guessing she got kicked out. Hello. Okay, you're back. I think something must have happened, eh? Great. Kiwi, are you there? Yeah, that's okay. You're back. You're back. All right. So we got the recording up and going. So we're going to go ahead and get started. So we are going to be focusing on the lab. So just as a reminder, every Monday, the lesson that we do is actually for the lab of the week. So you're actually not going to get this until a little bit later. If we take a look at our plan page, and, we'll be taking a look at, you guys will be getting the announcements today in just a little bit. But if we take a look at our pacing guide and if we take a look at our plan page, we're going to notice that this lab is actually slated. So I'm going to actually move this over so you can see exactly what I'm seeing. And that's over here. We've got the lab of human digestion is actually today. So this is perfect timing. So we're going to go through that lab because it's a little more challenging than our last couple of labs. Our last couple of labs, we spent a lot of time, going ahead and focusing on discussion posts. This one is going to be the first one where we're going to have some sort of lesson lab report, or at least questions that we have to answer. So let's talk a little bit about the objectives of this lab. So you just learned about digestion in general, and you learned about human digestion, which is actually really cool if you think about this. Our bodies have been doing this our entire lives, and now you're going to get to know a little bit more about what's actually happening. So in this lab, you're going to be able to explain digestion and waste removal in our organisms. So different types of humans, animals in general kind of go through this. We're going to have the opportunity to describe the process of digestion in humans using a specific anatomy, right? Using specific organ systems and identifying those organ systems, and then able to identify the individual functions and sites of secretion of these digestive enzymes. So knowing what the role is of the different organs and where these different enzymes and like chemical properties, like where does the hydrochloric acid in our stomachs come from? Where does this come from? We're going to have a chance to learn about that. So to begin, we're going to go a little bit through what the process of the digestive system is. So there's a couple of things that you might want to be writing down for this. And if you're watching this as a video, please feel free to pause and rewind as necessary as we go through each one. So we're going to go in order of the digestive system, where it starts and where it ends. And yes, my guess is you know exactly where it ends. So we're going to start with where it starts. So amazingly enough, the digestive system doesn't start when you swallow your food. Actually, it starts when you start to break down the food. When you first take that bite of that delicious apple, you sink your teeth into it. Remember that we are born omnivores. So the teeth that we have evolved for are both to eat plants and break things down, but also the canines to go ahead and tear whatever it is we need to bite into. In this case, my example of the apple, we use our canine teeth to go into the apple, and then we use our herbivore teeth, our molars, to grind that up. So not only, that's perfect analogy and example then, Kiori. Thank you so much for doing, not even realizing what you were doing for me. Thanks. So when we break down the food, we also have these things called enzymes that specifically are named amylase. And amylase is a specific enzyme that starts in your mouth where it breaks down the food. So you're already digesting that food before you even swallow. This is the same thing that when you think of something that tastes really good and your mouth starts to water, it's actually your glands producing that amylase that's getting in preparation of breaking down that food, which is actually amazing. It's what with like Pavlov's dogs, if you've ever heard of that experiment with the bell, you ring a bell, it is the dogs associate that with food, and then it starts that salivatory gland to produce that amylase. So that's the first part of the digestive system. So before you even swallow and finish chewing your food, you're already breaking it down. Then we go into number 2. So we go from the mouth, and then we move into number 2, which is right here in our esophagus, which is very different than your trachea. So really pay attention to that. Yes. Now, what I want you to pay attention to is the esophagus is the tube that you swallow food and it goes down into your stomach. The trachea is where we're breathing. That's how we get oxygen into our lungs, which we're going to talk about in the next couple of weeks. But specifically in the digestive system, we're using our esophagus to swallow using those muscles, right? So the pharynx right here is part of that muscle. It's what you use to swallow, pushes that food down your throat into the esophagus, and then it goes down to your stomach. So the stomach is over here, which corresponds to number 3. Once you swallow, your food is now sitting in your stomach, where your stomach is actually given off hydrochloric acid and enzymes called pepsin. Now, the pepsin and the hydrochloric acid is what is used to break down the food, specifically proteins, okay? And then the acid in your stomach kills a lot of the germs. So if you are eating something that has germs in it, it usually can't survive that really high acidic environment. So it kills off a lot of them, of course, depending on the germ or the bacteria or virus that you ingest is going to determine how you, how you feel and how that goes. Sometimes you can get an upset stomach from certain foods because of what this or the other. So that hydrochloric acid, however, is really important in breaking it down. And there are specific cells that there's like this mucus line that that lines your stomach that protects the cells. That's why we are able to have such high acidity in our stomachs without discomfort. Now, you may have something called acid indigestion, which means that that acid from your stomach starts to climb up your esophagus, and that's kind of when you get this really irritating burning sensation because there isn't that mucus lining in your esophagus like it is in your stomach. So just number 3, the important thing to do, actually, yes, kind of. You might be getting a little into that in some of the lessons, Kiori. You asked if we ever covered tapeworms or parasites. A little bit into this digestion, I did take out a little bit of the lesson that talks more about it. But only because of, pertinence and trying to lighten the load this quarter. But if that's something that you're interested in, Kiori, we can definitely explore that further in like the small group or something. So we go into our stomach, the acid kills the germs, all that. Once the food enters our stomach, it then goes to number 4. Our number 4 is then it moves into the small intestine. So our small intestine is actually this guy right in the middle. It's surrounded by the large intestine, but it's the small intestine is the one that's all that. It's all these big long tubes that get coiled. And that's because the food is going to move into that small intestine, which is then going to absorb the nutrients and transfer them to the bloodstream. So there's a lot of cilia. Cilia are those like long little hairs. They have like this tail-like feature. That lines up the small intestine, and the entire purpose of those cilia hairs is really to absorb those nutrients. That's why when you eat really healthy foods, you're going to feel different because those are different nutrients. You're getting a lot of those nutrients that helps your body with functions, what helps your body with digestion back into that circular loop. If you eat a lot of food that lack nutrients, you're not going to be collecting as much. You're not going to be putting that back into your bloodstream that then circulates throughout your body. You're going to be excreting that. And that's going to go from the small intestine. Whatever nutrients aren't absorbed, it's then going to be put into, it's going to keep there, but then your liver, so moving into number 5, so your liver right over here is then gives off bile salts into the small intestine to help break apart fats in the food. So that's when your liver is going to be used to break it up even further, really breaking down those lipids, right? We know that fats are a type of lipid. Lipid are fats. Oh my goodness. So it's going to be breaking that fat down into those food, into further, so you can get some more nutrients. So that's what the gallbladder does. And like I said, that is like right over here. Pancreas. The pancreas is over here. Excuse me, right here. And the gallbladder stores bile from the liver and secretes it into the small intestine to help break down fat. So I stand corrected. We're not talking about the pancreas. My own numbering confused me. But the step after the liver giving off the bile is that the gallbladder then stores the bile from the liver and then secretes it into the small intestine to help break it down. After that, we then go to the pancreas right here, which gives off more enzymes into the small intestine, which breaks down food and adds a solution to neutralize the stomach acid. So once you get into the small intestine, everything is broken up and is increased as well as you can. So that there is a neutralization. Remember that, and actually we haven't gotten to it yet, but there's a little bit about neutralization that we're going to look into in the next couple weeks. We're going to create our own solution to how to flatten out or make a very acidic environment neutral. And we think of neutral as water. Water is our baseline neutral, where there is no acid or base. It's just, normal, neutral. And then carries the gallbladder is directly under the pancreas. Yes, I know. I also confuse myself all the time. So appreciate your patience. And then the last thing that happens is the leftover products go to the large intestine where water is removed. And then bacteria help in producing and absorbing vitamins. And then once your food or everything that was taken out, all the nutrients, the water kind of gets sucked out while in the large intestine. It's like, and then you're left with solid waste or poop. And then you poop. And then you urinate. And that's your digestive system. So when we're talking about our digestive system, we want to understand exactly how the saliva is going to affect your breakdown of starches in this lab. So in this lab, we are going to need disposable gloves and goggles, which comes with your lab kits. You're going to need iodine, which is also in the lab kit. You may need a measuring spoon, cup, or bowls from your own kitchen. You'll want some sort of watch or something to take time on, and you'll want crackers. And if you don't have crackers, then any other type of starchy food. So crackers are ideal just because they have like a neutral color, so you'll be able to see the iodine show up a little bit more clearly. But you can do anything like fish, the goldfish would work, Cheez-Its would work, tortilla chips would work. A potato would work because it has starch in it, but it may just take a little bit longer in regards to how long you keep track of your results. So that's what you're going to need. Now, there is a level of safety in this. So a couple of things that you'll need to do in order to prepare for this lab. So this is today. So you're going to want to technically it's today, right? So you're going to want to be straightforward and you're going to want to be careful. Wearing clothing that you're not going to be worried about staining, because iodine does stain clothing. It can stain your skin for a period of time. It will eventually come out, but it can stain it for a little bit. Work surfaces as well, it stains. You're not going to want to be eating or drinking anything just because of the nature of this lab. You don't want to mix anything up. You don't want anything splashing into the food that you're about to eat. So making sure that you're not eating or drinking during this lab. You're also going to want to tell everyone in your household that you're performing a lab. If you have siblings or parents that are grown-ups that don't know you're doing it, they may not notice the iodine, they'll eat it. And then that's not going to be good for your lab. It's not going to be good for their digestive system. Therefore, make sure you tell everyone about that lab. And then the last thing is just know that iodine is what we're working with. It is heavily used and commonly used as an indicator, and it is toxic if you eat it or inhale it. And then it can also cause burns if it's too direct or too close to your skin. So make sure that you are being very careful with that. Okay? So this is going to be a little different than what you've done before. And that's because other labs that we have done have been pretty much cookie cutter labs. You know, they give you the exact directions. They tell you step by step what you're going to do. This one's a little different in that you're the one who gets to create this lab. So it's very inquiry-based, it's very self-directed. The most important thing, though, is that you document every single thing that you do. So that's going to be the big thing to keep in mind. So you're going to have a choice between two options for your lab. So remember, when we're thinking about the scientific method, we're really thinking, and let me see if I have it on here. I may have put it up on this. No, I did not. But remember that the scientific method usually starts with some sort of question. What is it that we're trying to figure out, right? And then after that, you do your initial research. Then you write your hypothesis, what you think is going to happen. Then you do your procedure. You conduct and do the procedure. Then you record your data. You analyze your data, and then you report your findings. So that's kind of, in a nutshell, how the scientific method works. So we're going to start with a question. You are going to choose between two questions. So two things, one of which you are going to want to look into and find more information on. So the first option is how pH affects starch digestion. So if you're not familiar with pH, pH is just the level of acidity or base that something is. So when we're talking about acid, we often think of lemon juice, vinegar. We'll often think about, tomatoes are very acidic, so forth. When we think about bases, we think about things like bleach, right? Bleach is something that neutralizes acids. We often use it in our cleaning supplies. So that's going to be a pretty big base. And so you're going to want to see, and so if you're choosing the option, how pH affects starch digestion, then you might want to think about adding vinegar or lemon juice to your saliva. And we'll get to that in a moment. We are going to be using your saliva, which is that amylase. That's where you get your amylase production. So you're going to have a chance to see how amylase activity is affected by the acidic conditions. So you'll take a look at how the environment of an acid environment directly correlates with the breakdown of starch from your saliva. Now, your second option is to check out how long it takes for starch to be broken down by amylase. So again, you're going to have a saltine cracker or a cracker. You're going to put it into the same amount and same mixture of saliva. And we'll talk about what that mixture means in just a moment, but for different periods of time. So maybe one is for 60 seconds. Another one is for 2 minutes. Another one is for 5 minutes, but everything is staying the same except for the time. So we're going to talk about that too. Remember that keeping the variable, one variable is changing, everything else needs to stay the same. So if you're doing the pH, the environment, then the only thing that's changing is what you mix your amylase with. The same amount of liquid is going to be the same, but your actual solution or the liquid that you add to your spit, your saliva, or your amylase is going to be the same amount of time, same amount of iodine. It's just the environment that's changing. Time is the only thing that's changing. You're going to mix the exact amount of amylase with water, most likely, the same amount of liquid in each test tube, but a little bit different in regards to the time for each one. So keeping that in mind. Okay, the general procedure. So this is going to be, it is already given to you in your slides into the document that you're going to download. This is the general procedure, okay? But you really should be adding specifics or changing your experiment based on the question that you have chosen. So something to consider is you are not going to be copying and pasting these procedures in your lab, right? So there is nothing that should be exactly the same. And don't forget that I do have that turnitin.com that identifies plagiarized work, and that does include work from FULET. So making sure that you are not copying and pasting the procedures directly into your lab report. They must be specific to your experiment and written in your own words. And QA, this is a lot of fun. So get excited and get pumped. So couple of things to note, okay? When we're starting, you're gonna wanna use a small piece of the saltine cracker or whatever starch you're using as a sample test. So you're not gonna wanna actually, so you're gonna wanna choose a small amount. This shouldn't be something that takes up half of the thing. We're trying to make a simulation, right? So trying to make a simulation of what is actually happening. So once you have a small piece of your cracker, you're gonna break up the saltine into smaller pieces to simulate chewing. Now, you don't want to actually chew it for a couple of reasons. One, it's kind of gross. Don't think that's very, doesn't look nice, doesn't feel nice, especially if you have to leave this out for a little bit. And also, if you actually chew it and spit it out, you're not going to be able to have, it's going to affect your controls because your amylase will already have started digesting the starch. So it's not going to be a true simulation. So instead, you're going to break it up into smaller pieces and put it into the test tubes. You're then going to dilute, and then you're going to spit into your container. So you're really going to want to make sure that you're spitting the same amount of spit into your containers. If you realize that you are running out of spit by your third test tube or fourth test tube, this sounds a little strange, but if you have a rubber band that's clean, you can chew on the rubber band because that chewing activates your salivatory glands, which then releases more saliva. So then you can spit into it. So that's just a little quick trick just in case. Okay. You're then going to dilute your saliva. So you're going to keep the volume consistent between samples, right? So again, remember, we are going to want to make sure that we are keeping the same amount of liquid. So if we keep volume consistent between samples with and without amylase, may dilute saliva with water. So for example, if you are doing the question number 2, where you're just doing the same amount of time, you're going to want the same amount of saliva to the same amount of water ratio. And you should be diluting the saliva with water if time is what your variable that you're testing is, right? Now, if you're doing the different environments, you're going to want the same amount of saliva and then the same amount of each different acidic or basic solution or liquid. So if you're choosing vinegar and lemon juice and then water and then, I don't know, something else, you're going to just want that to be the same amount of liquid total and the same ratio if you can. So that's something to consider. You're going to want to use tablespoons, or there's also the graduated cylinder that has been sent in your lab kit. Now, using the graduated cylinder is another science skill. If you remember from middle school, you should have had some experience, probably in 6th grade, about how to read the meniscus. The meniscus is that little kind of horseshoe shape or U shape that when you look through the test or the graduated cylinder, that it's going to kind of dip down. The meniscus, you're going to want to measure, you're going to want to get down to eye level and then measure the bottom of the meniscus. So that dip, that's where your amount is. It's the meniscus is where you're going to get the bottom line of the meniscus. It's like this U little dip that you'll see in the graduated cylinder. If you read it from the bottom of the meniscus, that's the actual volume of liquid that you have. So just a couple of tips when you're trying to measure this to avoid any sort of mistakes or any sort of variables out of the one that you're testing. Okay, once you've done all of that, you are then going to add the iodine only when you are ready to test the results. Okay, so if you are doing the environment, you're going to want to add the iodine. You're going to want to have all the same amount of times for the environment test, and you're going to want to add the iodine to the test tubes at the same time, or like one right after another, and then time it for the same amount of time. So whether you're letting the iodine sit there for a total of 30 seconds before you start recording your data, that's got to be the same amount of time for each one. Now, if you're testing time, let's say you want to test how the starch has been broken down after 30 seconds, a minute, 2 minutes, 5 minutes. You're only going to put the iodine into the test tube once you're ready to record your results. So 30 seconds, your 30-second test tube, you're not going to put the iodine until the end of the 30 seconds, the minute, after 2 minutes, after 5 minutes in each one of those. And then you are going to want to wait the same amount of time of the iodine before you record your results. So if you drop it in the 30 seconds, you're going to wait 5 seconds to see how it responds. That's fine. But then you need, after you drop the iodine after a minute in the minute test tube, 5 minutes, you're still going to want to wait 5 seconds before you record the results. So again, trying to only test it once you're ready to record the results, and then allowing that iodine to work with the substance for the same amount of time before you actually write down your results. Does that make sense, Kiori? Did that make sense? Okay, cool. And last but not least, don't eat anything. These are samples. They've already been exposed to iodine, and it's not a good idea to eat your tests. So again, remember, these are the general outlines. So a lot of questions that I had from last quarter was, how much is my volume? How much iodine do I put? And again, you are the one who is designing this experiment. So it's really up to you how much you want to put, right? You get to choose the volume. You get to choose the amount of iodine. I would say probably no more than 5 drops and probably no less than 2 drops of iodine. And you can make that more specific if you want. If you want to like measure the amount of drops instead of just counting the number of drops, that's totally fine. The volume, I would say no more than halfway, but you do want to have enough volume where it covers your starch. You're going to want enough volume or liquid to cover the amount of cracker that you have in there. So just those are my little kind of tools, my little rules of thumb, rules of thumb. Yeah, something like that. That's my tip for you guys when you're making your procedure. So just remember, that's the general one that you're going to do, but change it and make it specific for the experiment that you're doing. And you are writing a lab report, so you are going to want to explain those steps specific enough where that any reader, anybody who's looking at your experiment, can replicate your design almost to a T. So you really are going to want to be specific with the amount of each one, the materials that you use, the time that you kept. That's going to be part of your lab report. So before you start your lab report, a couple of things that you're going to want to consider. What is your independent variable? So again, what are you testing? What are you changing? What you change is what you are testing. So the independent variable is what you have control over, okay? The dependent variable is what are you measuring? What is changing depending on the independent variable, right? So this is, if my experiment was testing sunlight, sun exposure for 3 plants from the same species, right? The time is my independent variable. I have control or excuse me, I take that back. I was doing, no, yeah, the time, the sun exposure. So how long they're exposed to the sun, I have direct control over, right? So I can choose how long I expose each plant for 5 minutes, 30 minutes, and an hour, let's say. I am choosing the amount of time. The dependent variable would be what is dependent or how I measure the effects of that sun exposure. So for example, let's say I'm exposing growth, the growth of the stem, let's say. So I am then going to measure the growth of the stem is going to be dependent on how much sun exposure there was. So again, I don't get to control the stem growth, but I am controlling the environment in which that stem is growing, and that stem growth is going to tell me which sun exposure is going to be best for growth, right? So that's what I'm measuring. Is the independent variable is the sun exposure, how much I'm allowing the sun, it being exposed to the sun or in the sunlight. The measurement I'm doing is the growth, which is going to change based on the amount of sun. And then what are your controls? So what stays the same? So to continue in my example, with my sun exposure in the plant, I would have 3 of the same exact plant buried in the same exact pots, right? Same spot, same exact size pots, same pots themselves, same soil, so same exact amount, same type of soil, same nutrients. I would water it the exact same time, the same amount, the same time of day every time. And I would use the same way to measure as well. So really just focusing on keeping every single thing the same except for the thing that you're going to be testing. Okay. Questions on this? The independent, dependent variables and controls. Any questions? Okay, great. Thanks, Kiori. Okay, so we've gone through the lab. You're going to conduct your lab, and then you're going to start writing a lab report. So there's 5 parts to a lab report, and they do have an example that's going to be really, really good. It's the lab report guide, and they give you a template with another lab report as an example. So if you were to go to your lesson, there is going to be there's going to be a section that says lab report, guided lab report. And that's basically going to give you a brief explanation of each part with an example. And if I remember correctly, the example is from surface tension and water tension, or properties of water in general. So what I do want you to do is make sure that you delete those areas. It's okay to leave in the example and the explanation of those specific sections as you're typing it, but before you turn it in, delete that. I cannot tell you how many times students did not delete the lab reports guidelines, like what was already written in there, and they just wrote theirs underneath, and it was really difficult for me to grade. I couldn't really follow because it didn't really make sense. Or I've had students that just resubmitted the lab report because they thought that it was the actual right one, in which case they got a 0 because it was not correct. So just make sure that you're paying attention to that guided lab report. But in short, what I have here are the different sections of that lab report. So here I've got the title. So what you're doing with the title is you're telling us what the lab is going to be about. Just as a rule of thumb, if you can't think of, if you want a general idea, generally it's going to be something about the independent variable versus the dependent variable. But your title is going to tell me what it is you're testing. I should know exactly what I'm going to be reading based off of your title. Then you've got your introduction. That's where you're going to be describing your principles, and it's also going to give me the purpose. Why are you doing this? Why does this matter? And that's going to give your reader, it's going to bring them in. And so right away, they know what they're looking for. They know what it is they're getting themselves into. So making sure that you're intentional by describing your principles or purpose, and then any questions that the lab tells you to answer is what you're going to answer in your introduction page. So there should be a slide in the lesson, the lab lesson in 3.13, I want to say this is, where you are going to, and let's just double check, 3.13, excuse me. And so in 3.13, there should be a list of questions that you're going to answer in your introduction. The next part is your experimental method. So this is how you're going to describe your experiment and what you did, and you're going to describe the controls, so what you kept the same, and how you measured it. So how did you record your measurements? And there should be, it should be so detailed that anybody who does it can replicate it. So I would really, as you're doing the lab, try to plan it out. It's always good to go in with a plan. But if you're starting to go through your lab and you realize that it's not exactly what you thought it was, you're not exactly following it to a T. Maybe there's a couple things that you notice from your initial procedure that isn't working right. You can feel free to go back and edit it. It's just that by the time you do it in your lab report, it should be accurate to what you did when you were actually doing the lab. The next section is data and observation. So then you're going to want to include a description of your results, including any graphs. I highly recommend that you create a data table before you start recording your data. And then that way you can insert your data table directly into your lab report and include a graph. Okay, so graphs really need to be corresponding to what you're trying to convey. So if we're trying to make comparisons between different environments, then a bar graph might be best for that, right? We're trying to do a bar graph. Bar graphs are used specifically to compare different variables. So that would be really good if you're comparing the environment to one another and how much digestion or how much starch was readily available. If you were doing time, then maybe created, oh my gosh, how dare that kitten steal a hand? And no worries, Carrie, no problem. But a line graph might be really good if you're relating the relationship between time. The longer an item or an object stays in amylase, the more it's broken down, right? So creating that relationship between time and degree of digestion or breakdown. Once you have your data table and you included your data and your observations, then you're going to discuss and conclude. So then you're going to use the questions that are provided in your lab and you're going to answer them. But it's really important to know that when you're answering a discussion and a conclusion, you don't want to just make it so that each sentence in your conclusion is like a direct answer to the question. You want to make it so that it's fluid. You want to make it so it weaves in nicely, that your answers to the questions are actually embedded into your conclusion, not the answers are the conclusion themselves, if that makes sense. So start to get really into the habit of writing your discussions and your conclusions by making it sound like you already knew what the questions were already going to be. And so here are my answers. When somebody's reading your conclusion, it shouldn't be just like answer to question number 1. Then the next sentence is the answer to question number 2, right? It doesn't really make sense that way. And we are trying to practice writing a lab report. So just be mindful that you are trying to explain information to your audience, your reader. So there are going to be 3 questions that you're going to answer in your introduction. Again, you really want to make this more informational to your reader, not just straightforward, like I was told I have to answer the question, so here is my answer, right? So what is amylase is going to be one of your questions. How can iodine be used to monitor starch digestion? and what was the purpose of your experiment? So these information, these two that you start with, what is amylase and how can iodine be used to monitor starch digestion? Those answers are going to be coming from the labs that you read that, according to the pacing guide, were supposed to be completed on Friday. So those lessons that talked about digestion in general and then human digestion in the second one, it's going to be based off those labs. And there are some note-taking slides. So there's some slides that you should be taking notes on prior to starting this lab. That's going to help really identify the answers to these two questions. So go back, if you're having a hard time understanding why are we using iodine, go check your notes from the slides in this lab lesson and from your other two lessons previously. So if this is 313, so in 311 and 312 that you completed on Friday. And then last but not least, what was the purpose of your experiment? Remember those objectives from the lab. Why are we doing this? And why is this important for us to know? On the other end, you're going to have some questions in your conclusion. Write an evidence-based statement that summarizes the findings of your experiment. What did you use for controls in your experiment? What are some sources of error? And how could you modify or improve this experiment? So recall that in your conclusion, you do want to include the answers to these questions, but you don't want your conclusion to be the answers to these questions, if that makes sense. So for example, I would not have my conclusion be the evidence, the findings of my experiment were blah, blah, blah. The controls for my experiment were the same amount of liquid in every one, the same amount of amylase in every one, and the same starch used in every test tube. The sources of error are, right? That sounds not fluid at all. It sounds like I'm writing to a script. So making sure that your conclusion is fluid and connects sentence to sentence. All right. Here is the rubric for grading these lab reports. There are 4 main sections. Your introduction is going to be 25% of your points. So if there are 20 points total, 25% of that is 5. So 5 points are going to be your introduction, making sure that you include a clear description of the principles and concepts related to the experiment. The experimental method is going to be worth 5 points, making sure that you include a clear description of how the experiment was conducted. When applicable, describe the controls used in the experiment. 25% data and observations, provides data and observations that are easy to read and interpret. If data, tables, and/or graphs are created as part of the experiment, they are appropriately labeled. And 25% of it is discussion and conclusion. So answers the questions and making evidence-based claims about the results of the experiment, and cites data or observations and discusses possible sources of error. So that, in a nutshell, is going to be what you are doing for this lab. Are there any questions about this lab? Great. So that's everything I have for you. Feel free to get started on your lab today. It's going to be really, it's pretty clear. The hardest part is really going to be knowing which one to choose and then how to write that procedure and measuring everything to be the same. But once you get a handle on that, it's going to be really fun. I hope you enjoy yourself today. So other than that, that's everything I have for you. You can enjoy your wonderful Monday and let me know if you have any questions.